There is a mouse in a lab, and its cancer is glowing green. Nobody injected a dye into it. The tumor is making the light itself. A researcher is watching it on a screen inside a living animal while it happens. Here is how that same disease usually goes for a person. You get a scan. A scan is a photograph. It shows you where the cancer already is, which means it shows you where the cancer already went. Always after. For a long time, that was the only option anyone had. You could see the aftermath. You could not watch it move. What everyone wanted was a live feed of the thing while it was still deciding. That live feed exists now. It is sitting in thousands of labs, and no human being designed it. We took it off an animal. 1961. A wooden dock in Washington State. A man is kneeling at the edge of it with a bucket and a pair of scissors. His name is Osamu Shimomura. 16 years earlier, he had been working at a factory outside Nagasaki when the sky went white. He walked home through black rain. Now he is on a dock cutting up jellyfish, and he has no medical goal at all. He just wants to know why they glow. The animal in his bucket is the crystal jelly. You can read a page of print straight through its body. Around the rim of its bell sit more than 100 tiny light organs. Brush against it and it flashes blue-green. Once and then goes dark. Nobody knew how it did that. To find out, he had to cut the rim off by hand. Rim by rim. Animal by animal. The first summer, he and his colleague collected 10,000 jellyfish. 10,000 animals gave him a few milligrams of material, and the light inside it kept dying before he could study it. Then he gave up on one failed batch and poured it down the sink. The sink flashed bright blue. There was seawater sitting in the basin. Calcium was the switch. He found the trigger while he was throwing his own work away. So he purifies the protein, gets the light working, and reads the color. It comes out blue. The animal is green. Years of buckets and scissors, and his answer is the wrong color. So he goes back into the same rim and finds a second protein hiding there. In sunlight, it looks faintly green. Under ultraviolet, it glows bright green. He tries to make it react. Nothing happens. In his 1962 paper, he writes it down as green and appears to be useless. He kept going back to that dock for 19 summers, around 850,000 jellyfish, and the useless one turned out to be the one that mattered. Blue light goes in, green light comes out, no chemical, no fuel, nothing added. A bulb with no wires, which means it does not need the jellyfish either. Put that one gene into anything alive, and the thing glows. Nobody did that for 30 years. 1989. A scientist named Martin Chalfie is half listening to a seminar. Someone mentions a jellyfish protein that glows on its own. He sits up. To use it, you need the gene, the recipe. Nobody had it. One man was chasing it. Douglas Prasher at Woods Hole on a 2-year grant. He cloned it. Then the money ran out before he could prove it worked in anything. Chalfie tried to reach him. Someone answered the phone and said, "Marty Chalfie, never heard of him." Prasher hung up believing the man had quit science. The most important handoff in modern biology nearly died as a bad phone message. They found each other again and the clone arrived in the post. Other labs had already tried this and failed. They had cut the gene out with extra scraps of DNA hanging off both ends, and those scraps poisoned it. Chalfie copied the gene and nothing else. The dish went green. Then 2 nerve cells lit up inside a living, see-through worm. After that, Roger Cien pulled the rest of the colors out of it. Cyan, yellow, orange, and a red he named cherry. Now go back to the mouse. Tag cancer cells with the jellyfish gene and the disease carries its own light. You watch the tumor grow. You watch a single cell let go. You watch where it settles. A bone, a lung. In some cases, the glow shows up about a week before an X-ray can see anything there. A week on a disease where afterwards is usually all you get. October 2008. The Nobel goes to Shimomura, Chalfie, and Cien. It can only ever be split 3 ways. Douglas Prasher heard the news from behind the wheel of a courtesy van at a car dealership in Alabama. $8.50 an hour. Chalfie said they could easily have given it to Prasher and left him out instead. All 3 of them thanked him. 2 of them paid out of their own pockets to fly him and his wife to Stockholm. Years later, he was back in a lab. None of this was aimed at cancer. It started with a man on a dock who wanted to know why a jellyfish flashes, and we still do not know why it flashes. We can describe that light atom by atom. What it is for is still a guess. Most of the glowing jellyfish pictures you have seen are not even the glow. That is camera flash on glass clear skin. So what else is drifting out there right now? Doing something we would give anything to understand that nobody has thought to look at yet. Off the coast of Washington tonight, that same animal is still flashing at whatever brushes past it. It was never signaling to us. We just held it up to a lamp and took what we needed.